Okay, so I I want to share with the audience a little bit about some of work we have done, as we get some questions regarding rotational grazing and potential food safety risks. And in the last few years, we have done some on-farm and some uh, field trials on research farms, and I would like to share some of that data, um, or some of the findings. Um, and this is one slide from previous sessions that just remind that when you use and treat biological cell amendments of animal origin, and here we're referring to manure, and all manure can carry human pathogens, and some animals tend to be reservoirs for certain pathogens. For example, ruminants or cows are reservoirs for E. coli on 5.7, poultry, we see more salmonella, frequently salmonella and campylobacter. And when we use these biological cell amendments that are untreated, there is a higher risk since they haven't been in any treated to reduce pathogens. So it's recommended to prioritize the use of those in non-produced crops, although there is a common practice in certain regions with rotational grazing in crops, they are higher risk as vegetables. So what we do and how to reduce that risk, and one will be applying time interval between application and harvest. In this case, we use integration of animals, livestock, and you can see sometimes people will refer to rotational grazing, or they refer to crop livestock integrate production systems. So what this is that livestock or poultry, they will move from pasture or from a field to a field they'll be used for production, where they use either for consume the crop residues or cover crops. And while manures are deposited while grazing, they are sourced for fertility and organic matter, but also there is a potential source for pathogens, as we see for manure application. So what this requires a caution and planning when you use integration of livestock in produce to prevent cross-contamination. What growers are doing to try to avoid that potential contamination. So if they are part of a national organic program or certified organic farmers, they will follow the national organic program of 19 and 120 days rule for rotational grazing as well. So they will wait either 90 days if that vegetable crop is not touching the soil between the grazing event and the harvest for human consumption, or 120 days if that crop will in contact with the soil. We might consider there is less manure normal when deposited during the rotational grazing, and you might consider that's treated as wildlife intrusion. However, for the farmer, will be easier considering interval. And here on the side is just an example, one of the field trials we did with cover crops with sheep. So we did a series of field trials since there was not much data supporting the time interval between the grazing event and harvest of the vegetable products for human consumption. We did a series of projects, field trials to evaluate the persistence of generic E. coli and Shiga toxin E. coli in the soil. And after grazing sheep, and because the system we were interested in studying, they use sheep for the grazing. And this, I'm going to just show one of these initial studies that was done by my student, Laura Patterson, where we work with a working farm, a conventional commercial farm, and we follow the soil, the survival, the persistence of generic E. coli, and it's here on the graph, generic E. coli in the soil, and along of the time post-grazing. So, and we follow two fields, field A and field B, and because of coordination with the farm, one of the fields we are able to sample when the sheep leave the field, and another one later on. So basically, in general, what you see in this graph is identical what Dr. Milner presented earlier on. This is nice curve a peak and a die-off of that concentration of generic E. coli in the soil as the time passed and go through. And we saw that we are in particular interest on 90 days and 120 days because that's what farmers are following in terms of the NEOP. And you saw that between 84 to 100 days, there is a considerable decrease of the concentration of generic E. coli in the soil. And that peaks right after the grazing event and after some management practice as tilling of the soil, planting, change of type of irrigation, and so on. But then it's kind of a smooth and a drop of that concentration of generic E. coli. So another thing is this generic E. coli from the sheep? We know that is indicator of fecal contamination. So generic E. coli normal exists inside of the gastrointestinal tract of the animals, livestock, or they are actually related with the fecal samples. So to investigate that we did further testing some of those generic E. coli we found in the soil and also in fecal samples, so from the fecal samples of that sheep. And we're looking in fingerprinting, how genetically related are those generic E. coli and compared from the ones from the feces and the soil. And that's what this figure is representing. So what we identify some of those strains that were found in the fecal material and in the soil, they are very related, so they're kind of in a cluster at 80 days and so on. Something about this field trial is we don't have a control field. We don't know what happens when we don't use grazing. And that leads us to a second study on the same farm. And in this second year, we have a control field. So the fields they were grazed are here in yellow, green and orange or red. And the field that's controlled, not grazed, but still with cover crops. So the sheep were in the field when there is a cover crop. In blue is the control. So we see here that's a nice peak after shortly after the grazing and the tilling. And then by the day 90 that were interested and 120 days post-grazing, post-sheep left the field, actually there was no difference between the concentration of generic E. coli on the field compared between the grazed field and the control field. And another question is how likely, but we, as I said before, we still that generic E. coli is not pathogenic. So at the same time, we test for STEC, so it's a Shiga toxin E. coli, the same type of E. coli on 5.7, but there was no E. coli on 5.7. So initially we test the fecal samples. Do the sheep have STEC, and is that STEC potential survive in the soil? So in these graphs, this is here is not the concentration, but the proportion of positive samples we found in the soil after the sheep was grazing. And we observed that some of the between 50%, 75% of one of the fields of the soil samples were positive for STEC. That concentration really decreased and none were found after 90 days. Although we found a small percentage on the field B and field C, none of STEC was found. So we still focus in these 90 and 120 days. And what overall we saw is a decrease of generic E. coli by 19 and 120 days without no statistical difference between the treatment or between the grazed fields and the control. And after the 90 days, only a small percentage of the soil samples were positive for STEC. And as Pat mentioned in the beginning, one of the studies, we know they are variations of survival of generic E. coli with by year, by region, and so on. So later on, we were able to do a more control field trial, and now here in California, And now we expand that field trials to another states in Minnesota and Maryland to see these geographical variations. And here what I want to share is just some of the data of this third trial that we have different treatments. So we have a graze, so with the cover crops, and we have a graze, tilled cover crop, and fallow. And in this first year, and this field later on was used for was plant tomatoes. And here we were interested in looking is the difference between compost and non-compost fields. So part of the farmers, they will use a mix of compost and grazing. And although there's some complexity in this graph, but the bottom line for this initial first year of this project is, again, we're measuring genetic E. coli in the soil between the different three treatments. And only at the baseline, we saw a difference of that genetic E. coli between the fields that have compost and compost. And later on, at around 90 days as well, there was no significant difference between the genetic E. coli at around 90 days. Interesting or not, we in this compost field, we found that a spike around 123 days. And actually it was some outliers of the soil. So there's something going on in the soil, and these subplots were not graze, was actually fallow. So with this third study, we have a better control or more control experimental design, and we are looking more not just the food safety or the survival genetic E. coli and STEC in the soil, but also the colleagues from Dr. Galdin lab here at UC Davis. They are looking at the benefits on the soil health and on the yield as well. And then I mentioned before, this actually was, we are able to guarantee some funding to study in multi-regions and to see those variations that depending not just of the region, the weather conditions, geographically, and different managements as is between, for example, in California and Midwest. So the bottom line, when farmers are trying to comply with the produce safety rules, so the FSMA, which the following requirements they must follow for rotational grazing should be the NEOP rule of 90 days, 120 days, or treat as rotational grazing as the same as wildlife intrusion, and take the measures that are necessary to not harvest contaminated produce. Actually, the FSMA sees or treats the rotational grazing as a wildlife intrusion. So evaluate the risk and see of those areas and whether the produce has been safely harvested and take measures that necessary that the growing to assist the farm that during the harvest there is produce that is not potential contaminated. So again, there's another situation, there's different guidelines depending if we're looking at produce safety rule or any opinion specific of rotational grazing. With that, I know I get some of other questions, specific integration of crop livestock farms in orchard systems. And we actually were doing a pilot study on rotational grazing and orchards. So if any of the participants are interested here in California, so this is strict to California, would like to participate in the study or know more about, please contact myself or Dr. Rosie Bush. We can talk more about the study and potential collaboration. And that's what I have to share for rotational grazing. We still in this multi-state project I mentioned with cover crops and fresh produce, and we'll have more data findings and recommendations as we progress in this project. So we are in the first year. And next year we have different events actually for outreach and workshops as well that you can find those announcements on my webpage.